Welcome to Biology B. Our lesson today is 1.2. It's on gene expression and the lac operon example. The target of today's lesson is transcription factors. Transcription is the process that gets us from DNA to mRNA. Eventually, that mRNA would be converted into a protein, but that's another part of the protein synthesis process called translation. We're focusing on transcription, the first part. And these factors regulate which segments of DNA genes are turned on and transcribed or turned off, and then those genes are not coded for because they're not needed. Remember, a cell always strives to be efficient. They don't want to make proteins, especially enzymes that are workers, if there's nothing for those workers to do. You still have to pay them. Take a minute, pause the video, and read this little excerpt here. And take a minute, pause the video, open your guided notes, and watch this short video about lactose intolerance. Now that you've had a chance to come back after reading through here, I just want to introduce a few of the words. The gene regulation that's discussed is happening in a certain kind of bacteria called E. coli, Escherichia coli. It's a very common bacteria that's got a really bad rap. It exists in your large colon, your intestine, and you need it to help break down certain parts of your food. But it also breaks down sugars if you don't get it done in your small intestine. And lactose intolerance is what this whole thing is based upon. E. coli is good in your large intestine, but it's bad in other places in your body. But the focus of Jacob and Monod work is that this lac operon they discussed is used to illustrate regulation in bacteria. Gene regulation in prokaryotes like bacteria is a lot simpler than in eukaryotes, such as us, right? We are eukaryotes, we are multicellular. It's a lot more complicated. So we like to look at bacteria because it's easier. The success criteria for today's target is that we need to be able to explain that turning on and off the process of transcription can regulate gene expression. We learned a little bit about that in 1.1, but now we're going to look at it in much more closely as an example of the lactose intolerance situation. We're going to give an example of how gene expression is controlled with this lac operon. All right, just an overview of DNA to gene expression, DNA to protein. Remember that a gene is a segment of DNA that codes for, it's got a certain sequence of nucleotides on it, and when that DNA is coded, it's going to become mRNA, and then that mRNA will be translated into proteins. This step is transcription, and this step is translation. We learned about that in Unit 5 last year. I apologize, I'm not writing this very clearly for you, so let me just clean that up. So remember, I'll just recap there. A little segment of DNA is the gene, and when that gets converted to mRNA, that's transcription. That's the first half of protein synthesis. From mRNA to a protein, that's translation. So we're going to be focusing on this portion of the process. But in a normal situation, this gene is transcribed to mRNA and then translated to protein, and you know it because it's expressed by this, it's expressed in this quote unquote normal skin tone of these two individuals in the same population. So, for example, this is the protein that controls the amount of melanin in an individual's skin. These two are the same community, they're the same population of individuals. So the difference between them isn't that one is Caucasian and one is African American. The difference you're seeing is that this individual has a faulty genetic expression. So it's a faulty gene expression. We're going to learn about that here when we talk about albinism. But in a normal situation, you'd end up with beautifully dark melanin infused skin. But in the version of the gene that's defective in this situation, the protein enzyme makes it defective. I should say that the defective gene makes a defective enzyme, and that doesn't put the same amount of melanin in the skin. So the albinism is what you'd see expressed through this pathway. Remember that gene expression is the result of what you see in the organism. So we're going to focus, just one more time, we're going to focus on this portion of the process that's still happening in the nucleus from DNA to mRNA. That's transcription. That's why we're talking about transcription factors. There are also translational changes afterward, like insulin, but we are focusing on what's happening during transcription. Sometimes you got to trim up a protein and make it functional, but not in this case. We also talked about gene expression that gets out of control. So if the gene loses control in the cell cycle, remember the cell cycle is an interphase, then G1, S, G2, this starts with a metaphase, and then cytokinesis. At one point, if there's a defective gene or a defective section of DNA, then that's going to be called G0, where the cell that's defective would normally be killed off. It's called apoptosis. And in a normal cell cycle, that cell would no longer be allowed to continue into the division cycle. But in cancer, that cell's not removed from the process. So it is allowed to continue dividing. So you see your first mutation shows up here, the second mutation when the next division, the third mutation, the fourth mutation. All of a sudden you've got a lot of cells with the same abnormal, unhealthy DNA strands. So uncontrolled gene expression in this regard is called cancer. This should be animated in your guided notes. All right, the common example we're using as an example for faulty gene expression, again, is called lactose intolerance. So let's first introduce these players. If you see the letters OSE as opposed to ASE, that's not a typo. OSE refers to a sugar. So lactose is a sugar found in dairy products. It's a disaccharide, which means there are two molecules in there. You can see that over here in the illustration. Here's lactose. When it's broken down into glucose and the lactose, which is okay because we'll eventually convert it into glucose later. The enzyme that breaks that down is called lactase. That's usually how enzymes are named. This should also be animated for you to illustrate what's happening here, but you're seeing here's the lactose sugars. Then this is the DNA strand. There's a segment of the DNA strand that acts as a promoter, a segment that acts as something called the operator, and then there are three next to each other segments here that are all the three genes for the enzyme. These two and the repressor, these three players working together, are only there to determine whether these three genes will be expressed or not. We're going to learn about all these parts. But breaking down lactose intolerance, right? A person who is lactose intolerant doesn't break down lactose in the small intestine where other people do. If you're lactose intolerant, you're not producing the enzyme lactase, right? Which is happening here, which is why the lactose is allowed to travel to the large intestine. So this is a healthy gut. This is normal. In situation of lactose intolerance, the lactose is not digested in the small intestine. It travels to the large intestine where the bacteria E. coli are able to get to it. And when E. coli digest the lactose, they produce gas. They make their own wastes. They make gas as a waste and acids as a waste. And the body will bring in water into your large intestine to try to balance out all the chaos that's happening in here. So when you combine extra water in your large intestine, your poop chute, if you combine extra water, gases, and acids, you end up getting all the conditions that make what we know as lactose intolerance. All right, so the reason we have lactose intolerance is because some of our bodies aren't digesting it in the small intestine. So the lac operon, again, is just a segment of a DNA molecule, like a chromosome, gene. Here we go. There's a segment called the promoter. There's a segment called the operator. And then these three all code for some version of lactase, the enzyme. And we call this complex the lac operon. Okay, the lac operon complex is a series of molecules on the DNA strand that are going to eventually code for the production of lactase. We are looking at this in E. coli bacteria because it's easier for us to see their DNA sequences. Lactase, the enzyme, is only produced when lactose, the sugar, is present. If there's no lactose, then the gene is repressed. So there's no protein, no enzyme. Since they live in your large intestine, they live on whatever you eat. So if you drink milk, then you're providing E. coli with lactose, unless you break it down in your small intestine. A lot of people can't. So that's why a lot of people have varying degrees of lactose intolerance. The products, when bacteria break down lactose, is gas and what will eventually be diarrhea. When the bacteria do the work for us, sometimes that's more problematic. Now, there's a couple of numbers on here. We're going to walk through these labels. In this image, the yellow structure you see here, that's RNA polymerase. So remember that RNA polymerase is going to make a polymer of RNA that will eventually become the protein. That's the enzyme lactase. Now, on the DNA strand, on this DNA strand, you have something called a promoter, an area that's called an operator. There will be an inhibitor that pops in here. He's not on the strand, it's just a molecule that will bind, just like the RNA polymerase will bind, but it's not a part of the DNA strand. It's actually coded for farther down the line. And then 6, 7, and 8 are all 3 genes that will code for the protein, lactase. So those are the players involved. The promoter acts like a landing strip. It says to the RNA polymerase, this is where you start before you move down the line to code for these 3 genes. An operator will be ignored as long as the inhibitor is not attached to it. But I want you to think of an inhibitor like a roadblock. You can't cross. The RNA polymerase can't get around it. So it's effectively stopping the RNA polymerase from polymerizing the RNA needed to make lactase. If you think about it like a train track, this is where the train doesn't pass. Without lactose, now these are lactose molecules, sugar molecules. If they're not present, if there's no lactose up in this first scenario up here, the inhibitor will bind to the operator. When you ingest and eat lactose, that binds to the inhibitor and it distracts the inhibitor from binding to the operator site. So as long as there's no inhibitor, then the RNA polymerase can move forward and begin to transcribe these 3 genes. So as long as the inhibitor is not attached, then the RNA polymerase will be able to go through and transcribe those genes to make the protein. If there's no lactose, there's nothing to distract the inhibitor, and it'll bind to the operator, which stops the polymerase from moving forward. So how is the lac operon expressed and repressed? It receives signals from the environment. Eat milk, and that's one of the signals. Lactose present in the environment, in the gut, is a signal. Let's write that down. Lactose in the gut is a signal. It's turned on or off if there's lactose located in the environment. So if lactose is present, then lac operon is expressed and the enzyme is made, right? If lactose is, oh, that's a typo. If lactose is absent, sorry about that, then lac operon is repressed. It does not make the enzyme because the body a cell doesn't want to make something that it's not going to use. So please make sure we make this correction. If the lactose is present, then the gene is expressed and the enzyme and protein are produced. If lactose is absent, the gene is repressed. There's no enzyme made. This is what it might look like, another way of looking at it. In this case, here's the RNA polymerase. This is an enzyme, right? And it looks more like the enzymes we learned about in other semesters. It looks a little bit like Pac-Man. This is the repressor or an inhibitor, depending on some people use those 2 words interchangeably. Repressor or inhibitor. Notice there's no lactose here. If there's no lactose, the repressor binds to the operator underneath there, and the RNA polymerase doesn't have a place to land, right? It won't go forward into the DNA strand. So these will not be transcribed. So the repressor blocks the RNA polymerase, can't bind to the DNA, transcription does not take place, no enzyme produced. Okay. In this example, there's your lactose. Lactose is binding to the repressor or inhibitor. If there's nothing to, if the inhibitor or repressor is distracted, then the RNA polymerase will bind to the promoter because there's nothing on the operator. It just completely ignores the operator, moves forward, and what you end up making is this strand of mRNA. And then if the mRNA is made, mRNA will be transcribed to the eventual lactase enzyme. Here is a practice sheet for you guys to work through. It says when transcription cannot take place, what is happening? When transcription cannot take place, no transcription, the gene has been turned off. What happens when all the lactose in the environment is broken down? Well, your options are the RNA polymerase binds to the operator, but remember, the RNA polymerase is what makes the enzyme. So if all the lactose is broken down, there's no more need for that enzyme. The repressor binds to the operator to block transcription. Transcription continues. And if you're checking your notes, that's okay, but the answer here is the repressor binds back to the operator. So make sure you can answer these by matching the diagrams to this description here. You have operon genes, and there should be like another arrow here, but that's number 7. The operon itself, this whole structure, is number 6. RNA polymerase is the enzyme that's going to be doing all the work. That's number 4. mRNA is what's produced. That's 5. The repressor, in this case, is number 2. The regulatory gene is number 1. And the inducer, in this case, that's the lactose. That's number 3. It's inducing the process by distracting the inhibitor. Repressor. You will have a question like this on your test. Choose A or B for each of these. Transcription of lactase will occur, A or B. Where do you see transcription happening to make mRNA? I'm going to have you guys do this as homework. I'll walk through it with you. I'm not going to give you the answers. I'm going to come back another day as a warm-up, and we'll go over the answers then. Number 2, lactose is absent. Which of these do you not see lactose? A or B? Number 3, E. coli needs to make lactase. Needs to make lactase. Remember, this is the enzyme, so why would it need to make lactase? Only if there's lactose present, so A or B. The gene will be repressed and the gene will be expressed. Make sure you can answer these questions on your own because they're going to show up on a test. I will go over the answers to these in a future video. If you want a refresher for explaining what lactose intolerance is all about, what causes lactose intolerance, what happens to the body of a person with lactose intolerance, and how it might be alleviated, fixed, helped along with a pill, a lactase pill. So you can click on this button, and if you click on this button in your guided notes, you can watch this video and answer the questions about it. I'm going to have you do that on your own because you have access to that in your guided notes. The answers will be posted in the review, so you'll be able to check your understanding of that. So that's another portion of what I want you to do, just to follow through and do. Hate to use the H word here, but I want you to do some homework so you're prepared. Okay, it's kind of like studying. All right, that's all I've got to cover for this lesson. Make sure you can hit the learning targets to be successful, your success criteria for the day. And let me know early if you need any review.